Hi, this is Dr. Lauren Lanham from the Keene State College Department of Biology. I'm going to talk to you about the cell and microscopy pre-lab video. I'll cover the expected learning outcomes, the pre-lab assignment work, and give you an experimental overview, and talk about the post-lab assignment work. The expected learning outcomes are that you will be comfortable using a compound light microscope after you've completed this lab, including its basic use, things like focusing, specimen preparation, and microscopic measurement. We also want you to be able to do basic calculations involving microscopic units. We want you to know the structural distinction between eukaryotic and prokaryotic cells and develop a basic appreciation for the numbers and habitats of microbial cells. We also want you to be able to conduct and interpret a gram stain as used in applied and basic microscopy or microbiology. So for the pre-lab work, you need to read and print the lab handout, which contains the lab protocol and the assignment. And you also need to watch and take notes on the videos posted for this lab, which include a video on how to do a heat fix smear, a crystal violet stain, a gram stain, and how to focus your microscope. When you've done those videos and you've taken notes on those things, you should take the pre-lab quiz, which is available to you online. You should know by now that all life can be categorized into one of three domains: the bacteria, the archaea, and the eukarya or eukaryota. Microbial life is all that you'll find in the domains bacteria and archaea, and it's most of what makes up the domain eukarya. We also fall in the eukarya, however. We belong to the animals, and we are one of the multicellular groups, as are the plants. Now, if we were to look at all those life forms, we would see two basic cell patterns: eukaryotic cells and prokaryotic cells. And the fundamental difference between the two is the presence of a membrane-bound nucleus and other membrane-bound organelles. And this is inherent in the name. Eukarya means true nucleus, and prokarya means not a eukaryote. It's kind of a name in the absence of something else. The domains bacteria and archaea have prokaryotic cell structure, and the domain eukarya has organisms that all have eukaryotic cell structure. This is a typical prokaryotic cell. There's no nucleus. The DNA is simply anchored at a region called the nucleoid. This is a typical eukaryotic cell, and you can see the large nucleus, and it is actually wrapped in a double membrane structure. So the nucleus contains all of the organism's DNA enclosed in a membrane-bound organelle called the nucleus. So you're going to be using light microscopes in this lab, and light microscopes are useful for measuring cells. Cells are measurable in units called micrometers, and there are 1 million micrometers in a meter. And so you're going to need to get comfortable with that unit of measurement as you do this lab and as you proceed in your career in the sciences. An experimental overview is shown here for part 1, and in a minute I'll show you part 2. The first part of the lab is gram staining. So the gram stain protocol involves taking a drop of a bacterial culture, spreading it in a thin film over a glass slide, allowing that slide to dry using a slide warmer to speed the process, and then heat fixing the cells to attach them firmly to the glass slide. Once they've been heat fixed, you can then proceed to the staining steps. And in the gram stain, there are 4 staining steps. We use crystal violet, we wash it off, we use Graham's iodine, we wash that off, we use a decolorizer, which is not a stain but an acetone-based solution, to wash dye out if possible. And then we use what's called a counter stain, and that's referred to as safranin. The lab video will give you more details on that particular process. At the end of that staining protocol, you should be able to see whether a cell is gram-positive or gram-negative. These are 2 important cell types amongst the domain bacteria, organisms that belong to the domain bacteria. There's more detail on that in your lab handout. To gain an appreciation of the difference between prokaryotic and eukaryotic cells, you're going to make a culture, or you're going to make a microscopic slide that is stained from your mouth. And if you sample your mouth, you should capture some human cells, which are eukaryotic, and some of the many bacteria that are found living in your mouth. So to do that, you will scrape some sample off your teeth at the gum line, gently, and mix that sample into some water, spreading it thinly, let it dry, heat fix it again to attach the cells to the glass slide, and then simply soak that sample in a stain called crystal violet. The slide incorrectly says crystal iodine, it should say crystal violet. Let that stain sit for 1 minute, then rinse in DI water, dry the slide, and examine with a microscope. Once you've done your lab, then you're going to need to complete the post-lab assignment. Your instructor will give you a deadline by which that is due, and this will be announced using the Canvas system. Thanks, and we will see you in lab.